This is a production of Cornell University.
I'm Joanne Labate. I'm the Vegetable Crops Curator at PGRU in Geneva, New York. We're the Plant Genetic Resources Unit.
And my name is Zach Stansell, and I am the Industrial Hemp Germplasm Curator, although I have a great passion for vegetable germplasm. So we're really excited to show you some of our field increases this year. We're regenerating a number of annual and biennial crops, and we're really excited to show you around. So although the primary emphasis of this work is seed regeneration to replenish our seed lots for distribution, this work also entails characterization and just general germplasm and genetic diversity conservation. So welcome.
All right, so as Zach mentioned, we have biennial and annual crops. Onions are biennial. In the first year they produce bulbs, and then we have to dig up the bulbs, put them in the barn over the winter, and plant them the next year to produce seed. So first I'll show you the first year onions. They're here. We have 20 different types, or accessions we call them, of onion that we're regenerating this year. And the plots are laid out on these plastic strips. And we try to keep a population size of 50 to 100 plants per population so that we can get the random mating and keep the diversity. So I just wanted to show you our bees. We rent bees every year, and they're back here. We're not going to get too close because they're active because it's so warm out. But when we do the pollinations, each accession or each population is isolated in a nylon mesh cage. And these bees, somebody comes in early in the morning while it's still cool out, and they transfer these boxes of bees into the cage and leave them in there for a few weeks. And all they really need is water, and they feed off the nectar of the flowers and pollinate. I wanted to show you our second year onions. As I said, these are biennial. So what was planted, this is 2020, allium is onion, and plot number 108, and this is an accession that originated in Portugal. What was planted are the bulbs, and you can see the bulbs are kind of sticking out of the soil here. And these will grow the plant and they will flower, and before they flower, a cage will be put around them so that they can have the bees put in and be pollinated. But we'll let them grow up a little bit before we put the cage over it.
So here we have a cabbage increase cage. So this species is Brassica oleracea, the dog of the plant world, I should point out. It's a species that contains broccoli, cabbage, cauliflower, kale, Brussels sprouts, Chinese kale, etc. These cabbages are kind of unique in that they have a biennial life cycle. So these seeds were actually started last year, 2020, grown out 1 year vegetatively, dug up and stored over winter, basically in a protected but outdoor environment in upstate New York. So they got quite a bit of chilling hours during that time. So we moved them back out to the field and transplanted them, and they're just now starting to flower. So many of the materials that we have out here are not great cabbages. They're not something you'd be thrilled to pick up at the grocery store. However, that's kind of that's a feature, not a bug. These represent very genetically and geographically diverse materials that, you know, while they maybe aren't the highest in horticultural value, they represent some sort of local adaptation or potential disease or abiotic stress resistance. So from a plant breeding perspective, it's incredibly valuable to maintain these diverse Brassica resources. Regarding the uniformity, you'll notice that unlike a inbred line or an F1 hybrid population, the plants that we keep in these cages are not very uniform, and that's actually the kind of the main point, is to preserve this population level genetic diversity. This lack of uniformity is actually kind of a sign that there is some sort of segregation occurring in these populations, and so we try to keep a large enough sample size, 50 to 100 plants minimum, to maintain that, especially since Oleracea is an outcrossing species. Having a larger population size is really essential to maintain that diversity long term.
These plots are our tomato regenerations. Tomato is our most popular crop that we conserve, and we have about 6,000 different accessions of tomato. It's our most economically valuable crop also. Cultivated tomato is self-fertilizing, so this is the only crop that we regenerate that we don't use honeybees. We don't use cages, so they're planted in these rows, and we don't need as large population sizes because they are self-fertilizing. We just need enough plants to get a lot of seed. So we do 24 plants per accession for tomato, and we're doing 50 accessions this year. And you see the wooden poles that are in. The plants will be trellised when they get big so that the plants are upright, and that helps protect them from things like disease, and it keeps the plots from growing into each other, so we can make sure we have the identity of the fruit when we harvest it. Celery is another crop that we conserve. It's also a biennial, so it gets treated like the onions. This is our first year celery here. You can see the small young plants, and over here is the second year celery. So they've been in storage all winter, and they were planted. They have a kind of a big root on them.
So we brought you to one of our seed processing areas. So this is some of the equipment we use to thrash and clean our seeds before they're packaged for long-term storage in a -18 freezer. One critical element of maintaining all of these collections, there's a total of approximately 12,000 unique accessions spanning several hundred taxa. So it's really vital that we regenerate those materials periodically. If we do 100 accessions a year, then that's 120 years to flip over the entire population or the entire collection. So it's really important that we develop new collaborations going forth. Please feel free to contact Joanne or I if you have the capacity or willingness to help us with those. We'd love to have those conversations with you. So we have 3 primary core groups of stakeholders that utilize National Plant Germplasm System collection. So a primary user is plant breeders who, like I mentioned before, will find accessions that are locally adapted or have unique R genes or are valuable in some regard for their breeding programs. People conducting very basic plant science research will often request our entire collection to screen it to, for example, conduct association mapping for a certain gene. And then there's a lot of educational value in these collections as well. So certain higher institutions will use these materials. I actually pulled some of the Brassica collection to show a domestication timeline presentation at Cornell, which is a lot of fun. So your biggest limitation is your imagination when you have this much genetic diversity. Giving you a full tour would probably take the whole video session. Today we didn't include the asparagus collection, which I'm working on regenerating. We also keep many leafy brassicas. The radish collection is kept here. We have the buckwheat collection as well, and yeah, thank you, Joanne. The winter squash collection is kept here as well.
If you are a plant breeder or someone doing research or higher education and you're interested in obtaining seed or germplasm from the National Plant Germplasm System, we have a nice database, a web portal online called Grin Global. You go there, create an account, and you can order seed. We ship seed all around the world, and if there is any information about the accession, the passport data, That will be in that database, and you can find it on Green Global. If there's any images, same thing, you'll see it there.
This has been a production of Cornell University, on the web at cornell.edu.